startups, MSMEs, and industries. Through this series, we aim to showcase faculty innovators from multiple educational research institutes, featuring innovations across multiple industrial sectors. This platform is designed to forge pathways towards impactful technology transfer and dynamic commercialization opportunities. This is one of the mandates of TEC's responsibilities. To elaborate a little more about who we are and what we do, I would like to invite Professor Deepak Chitkara, coordinator of BITS Technology Enabling Center.
Yeah, thank you, Anchal. So I hope I'm audible to everyone. So can anybody confirm, Professor Anupama, if I'm audible?
Yes, you're audible.
Okay, great. And if you can just switch on your camera so that we can see you as well, it would be great. Okay, great. Yeah. So it's a proud privilege for me to introduce the BITS Technology Enabling Center. Just to start with, let's look at the genesis, that how it began. So this technology enabling centers have been created by the Department of Science and Technology. DST has a mandate of for the technology development and commercialization, and they are doing various activities around that. So establishing these TECs in the university system is a step in that particular direction itself. Now, one thing that has been seen there, which is missing, but we can say that if you look at the university system, that there is a lot of research that is happening. The patents are being filed, they are granted, but very few are leading to the commercialization. So to identify that where are the lags, that why they are not being translated, so TECs has come up and taken up one of that challenge, that why not to look into different aspects of it, bring in all the stakeholders at one place and try to understand the challenges which are being faced by the innovators at one end and the industries on the other end, so that the translation can happen smoothly. And whenever we talk about the translation, there are many stakeholders involved. It starts with the development of the technology by the innovators, the filing of the patents, protecting that intellectual property, then followed by pitching at different industries or to different stakeholders who can put in their investment into it. Then there are several steps of development, and we all know about these technology readiness levels. So it has to go stage wise, and at each step many other stakeholders are coming in, and depending upon the technology, the investment is also needed. So bringing everyone together at the right time is a daunting task, and there the role of TECs come into the picture. Now DST has created these centers in different states of India, and for the state of Rajasthan it has been awarded to BITS Pilani, and BITS Pilani with its partner institutions as well as other stakeholders have working in this particular progress. Like we have partnered with Vivekanand Global University, MNIT Jaipur, then Central University of Rajasthan, Maharana Pratap University of Agriculture and Technology, Udaipur, and then Bikaner Technical University. And beyond that also we are going to various colleges looking after that. And through our satellite centers at BITS Goa campus and BITS Hyderabad campus, we are further extending to these regions as well. So there are in all 22 technology enabling centers has been created in different states, and these also talk to each other. So if a technology is being developed in a particular location and its taker is present in a different location, they all could be connected together. So that's the overall system which has been created. Apart from that, other stakeholders such as the various Chamber of Commerce's, CII, like we have Rajasthan Chamber of Commerce with us, and various advisory members from the government bodies are also a part, as well as the IP experts, so that all the components could be seen together. So if we see for any technology to see the light of day, there are multiple steps, and in fact these multiple steep steps has to be taken. Now one of the initial step is to have the intellectual property in place, identified, filed, and granted patents, and followed by pitching of those technologies. Now the idea of coming up with TechMate series or episodes is to identify the faculty innovators who are working on various technologies, so that they can come forward and showcase their technologies with a possibility of future investments into these technologies, so that they could be taken forward. And when we talk about the investment is not only in the financial terms, but also certain hand holding or certain guidance or advisories, certain network, because it is not only the financials which are needed for a technology, but other components are also involved. So TechMate series is basically created with that mindset. So can we have identified innovations, and then we can ask the innovators to come and present their technologies, and then we can go forward with that. So we have categorized into different domains, and one of the domain which we are starting today is with the biotechnology and pharmaceutical sciences. And we have the innovators from our different campuses, like we have Professor Anasuya Ganguly from our Goa campus, Professor Swati Biswas from Hyderabad campus, and Professor Anupama Mittal from Pilani campus, who would be showcasing their technologies, and we'll love to hear further with them. So with this, I welcome you all for this event, and also I invite you to join the activities of the BITS Tech, and we are very happy to collaborate with all of you in our future endeavors as well. So with this, thank you everyone. Over to you, Anchal.
Thank you, Professor Chitkara. So, as he already mentioned, in our inaugural episode, we feature 3 faculty innovators from BITS campuses, Goa, Hyderabad, and Pilani. Being educators and researchers for years, these scientists have come up with brilliant innovations, tackling some of the most pressing challenges in the field of healthcare and biotechnology. So, starting with Professor Anasuya Ganguly. She is a faculty member at BITS Pilani Goa campus. She specializes in the areas of stem cell regeneration in creating a part or whole tissue engineered organs. She also likes to explore human genetics, cancer biology, ecology, and environmental sciences. Concurrent with this, her lab's research focuses on environmental health, regenerative medicine, and tissue engineering. They have investigated environment's toxins' impacts on kidney diseases, developed functional tissue from human stem cells, explored materials for heart valves, created microfluidic platforms for cardiac drug testing, innovated bioinks for 3D printing, and advanced skin grafting techniques. Professor Ganguly is a co-director of the company Backtreat Environmental Solutions LLP since 2015 and Tissue Engineering Private Limited since 2022. Today, she will be describing a small part of her research that she is very passionate about and envisioning the product development very soon. Let's hear more from her. Over to you, ma'am. Ma'am, you are on mute, I guess.
Yes, thank you, Achal. Yeah. So do I start with sharing the screen or do I introduce myself first?
Ma'am, you can share the screen and introduce yourself.
Great. Very good morning, everyone. Yeah, thank you for the wonderful introduction, Achal. Achal is also our ex-student, so that makes me feel very proud to have Achal on board. So good morning, everyone. I am Anasuya Ganguly. I am from BITS Goa. I am a professor at the Department of Biological Sciences, where I teach and do a bit of research. And we have 2 spin-offs from our work, 2 startups, as mentioned. So today I'll be talking more about TEPL, or Tissues Engineered Private Limited, and where I try to make miniature tissues using scaffolds from natural materials, cells, and different kind of biomolecules. So we registered in 2022, and we focus mostly on healthcare innovative products. And we are incubated in our BITS campus itself. We have a bio incubator called BITS BioRack BioNest, where we do our research and get all the help from the incubator. Right. So what kind of things do we produce and what kind of services do we give? So we have some products that are almost ready, especially tissue engineered skin patches. We develop different kind of bio inks for 3D printing. 3D printing is a really upcoming technology that everybody must have heard about. So where we add a gel-like material that helps as a scaffold with which we mix cells to print different kind of tissues, and we condition it in the lab. We also print different kind of tablets and drugs for patients. We can customize them depending on the dosage, depending on the preferences of the patients, etc. And as for services, we do biomaterial testing. We work with 3D cultures, and these can be developed for any drug testing, cosmetic testing, or we can even take them further into tissue engineering with the intent of helping people who might have some tissue malfunction. So it's part of regenerative medicine. So we can do that as well. As all of you are aware, nowadays we have strict rules for animal testing. Animal testing is almost going to be banned, so we need an alternative. All said and done, this 3D culture systems, cells, tissue culture, these are all a very good alternative to animal testing. We can particularly make a part or an organoid of a particular tissue that we want to test the drug on. We can make it in number of units, so we can test it multiple times. Then also we can test cosmetics on engineered skin instead of testing it on animals or even on human subjects. So this is a wonderful alternative that we are exploring, so that current products that will be developed need not go through all that animal testing, need not be so expensive also in the testing, and we can get into the market much faster. So this is the value proposition. So we want to develop 3D bioprinted skin tissue construct for drug delivery, for drug testing and cosmetic testing for the global market using novel biomaterials. So the technology is very simple. So if you have a bioprinter, you just load it up with bio ink. We have to standardize the bio ink before, so we have to test it out so that its fluidics is perfect, so it does not get stopped while printing and all that. Viscosity is very important factor. So we make the scaffold or the material as a hydrogel, as a viscous material, and then we can add cells to it. We can even add different kind of medication or biomolecules. And we print. We can 3D print into different patches or scaffolds. And depending on exactly the shape that we want to generate it, we can do that in a 3D printer. So that is the good thing about 3D printing. Alternatively, we can even do it manually in the lab in Petri plates. What 3D printing is, if you have a skin injury, which is of a certain depth, then you can exactly fill it up. The shape will be perfect fit. So that is the main advantage of using a 3D printer. So it is animal-free biomaterials. We use plant-based materials. We also use different kind of PCM molecules. And these all make it, if you are making it in India, much more cheaper than what is available in the market. Many a times we have to import different kind of skin patches and all that, and those are very expensive. And if we are 3D bioprinting it, the consistency is very good. Plus, we can customize it depending on our customers and patients. So currently, we have made one in which we use scaffolds made from corn husk. Now, these are basically little bit of definitions about tissue engineering. I already told you, you have to have scaffolds, cells, and biomolecules together, and we can make it into a small functional tissue construct. that can restore, maintain, or improve the functions of an organ. And I will tell you more about the particular corn husk scaffold in a minute. So this is an overview or a pictorial slide to explain the whole methodology. Natural materials, plus we also use pharmaceutical polymers. The good thing about using pharmaceutical polymers is they're already approved, so they have all the regulatory, certifications. So it's good to use them. So we use them also, and we use in different concentrations. We make different composites. We cross-link, then we characterize them for their, mechanical strength, chemical properties, and how they are doing biologically. Like, are they biocompatible? Are they okay? They are not toxic for the body, etc. So after doing all these tests, we mix them with cells and we bioprint them. And the type of cells, like currently, since we are interested in skin tissue engineering, we add different kind of skin cells, fibroblasts, keratinocytes, etc. And then we put it back into the incubator with media, and we characterize them after some time if they possess all the properties of that particular tissue that we wish to generate. So coming back to the innovation, so scaffold materials are very important for tissue engineering. And we thought, why not use corn husk? Because people just throw it away, and it seemed like a good candidate. So we went ahead and we decellularized it, because cells normally help in eliciting those response. So we cleaned it up, we decellularized it, and we used the basic skeleton of the corn husks to grow cells on. And the results were very promising. So if you see the first two pictures, scaffold without cells, so these you can see even the tiny holes. These are the stomata in the scaffold. These are scanning electron micrographs. And in the next two panels, I have two different type of cells that I had added onto the scaffolds. And the cells seem to like it. So they are getting attached to the scaffold, the corn husk background, and they're growing on it. Their morphology is pretty good. So these are the results from our corn husk used as a scaffold material for tissue engineering. We further stain the nucleus of the cell too, and we can see several nuclei. In the background, you can see this brick-like structure. That's the scaffold material of the corn husk. And the tiny, tiny blue bright dots are the nuclei of the skin cells growing on it. So we could say that these are good scaffold materials for tissue engineering, can be used with different kind of cells to generate, miniature tissue constructs. And since it's a natural material, so a lot of positive advantages to that. It has less chances of getting rejected, and we can make many different type of tissues with it. Coming to the possible market size and competitive landscape of this product. So in general, global reconstructed skin models or skin tissue constructs market was valued at US dollars 37.1 million in 2023, and it is growing at a compound annual growth rate of 14.6 from this year to 32. And if you want to talk about the competitive landscape, today it's there are a lot of global players in this field, but most of them are abroad. So there's one called Skin Ethic, Re-Epi-Skin from France, Epiderm from MatTech USA, StrataGraft from USA, etc. And there are lots of similar aggressive players who will come up in the next 5 years, all from United States, like StrataTech, Avita Medical, BioTissue, etc. But as you can see, in India, it's still to pick up. As for global 3D market, there are lots of players there too. We have some regional players, country niche players, and global players all in our pie chart estimation in 2021. There are a lot of companies, but as you can see, none from India. Pandorum is a very famous one that is coming up. Regenerative, Inventia, Vico, etc. Organovo. Keep hearing these names. So what is the social relevance and impact of this technology? So first, we can eliminate the need for animal-based biomaterials. We can have bioprinted skin tissue constructs that the pharmaceutical industries and the cosmetic industries can safely use to test their new drugs and cosmetics as alternative to animal testing. Since that's kind of like the mandate for the regulatory bodies nowadays, so we can, do away with animal testing for biomaterial isolation and preclinical studies. As for the commercial potential, due to ban on animal testing, in vitro testing methods like organ on a chip, organoids, 3D culture systems, bioprinted tissue constructs are gaining popularity for testing new drugs and cosmetics. And I told you about the market demand for skin tissue constructs is growing. Expected market size will be around 4.52 billion by 2030. So at the bottom of this slide, we have a summary of what I just told you. So you can have cells, you can mix it with hydrogel, bioink, scaffold-like material. We can print it out and we can use it for testing. And there'll be no need for animal testing as a result, as a consequence. So when it is done, then we can analyze it, we can test it for different kind of properties, and we can take it to the market. As we were talking about TRL levels, so we are kind of at a TRL level of 3. So we just have like a proof of concept kind of thing. So now we have to go into our testing phases. Then we need from regulatory bodies like FDA, etc. And finally, we can go into scaling up and commercial production to take it to the market. So traditional time, it takes around 10 to 15 years. So at the stage that I'm confident that in 3 to 4 years, we'll be able to finish it off and take it to the market if it goes well. So this is the business canvas model of our startup. So we have partnerships with a 3D bioprinter company called NBIL in Bangalore. Then there's a company in Armenia called Foldink. So there are a bunch of doctors. They made some very specialized bioinks that they would like us to test. So we are working with them. Then there's a similar company in Bombay called Core Additive Manufacturing. So they are also into 3D printing. So we are also working with them for different kind of materials. So our key activities are tissue engineered products and 3D printed organoids for drug delivery, for drug testing, for cosmetic testing. And we can provide services in biomaterial testing, training, etc. So our value propositions are low cost. We can customize the 3D bioprinting, and it's animal-free, regulatory approved biomaterials like pharmaceutical polymers, and we can scale it up. So our basic revenue stream is business-to-business product sales, technology transfer, trainings, etc. So we are interested in increasing partnerships and collaborations with different organizations to take our product to the market faster. And our target customers are mostly pharmaceutical and cosmetic industries, and also the medical healthcare system, where burn patients need tissue constructs to help them heal quickly and for better outcome of their regenerative skin. So after we are done with our products, we would like to do more testing, more validation from different companies, from different markets, get regulatory certifications to take it to the market before we can scale it up. So we are looking for licensee and our partners for development of our product. So we have a lot of interesting people in our team. So I had a lot of interns before, but currently Ajita Sharma is the intern working in our incubator. And myself and Professor Shrikant Mutnuri, we are the co-founders of our company, TEPL, or Tissues Engineer Private Limited. So these are some of the people that I worked with or still are working. So here is Maheshwari. This is another team who had a bioprinter, so they came and helped us out. Then Hemant is a PhD student. This is Ajita. So these are some of our more work. I was talking about the tablets that we could customize and print for the patients. So using different kind of materials. This is a charcoal tablet that absorbs toxins from the body. And this is another scaffold with cells growing on it. We have filed for 3 patents, and we are waiting for it to be awarded. One is on composite materials, pharmaceutical polymers. The other one is from corn husk, and another one we did from tomato skin. So we got a little bit of help from BioNest, just like a startup seed grant. And we also got a little bit help from AMTZ in Visakhapatnam for a bioprinting project. So thank you all for listening to me and about my work. I would be happy to answer any question that you might have.
Thank you, Professor Anasuya, for a wonderful presentation.
Thank you, Anshul. Thank you, everyone.
The floor is open for questions. Audience members, if you have anything to ask or say, please write it in the chat box. We'll interact with the innovators on your behalf.
So meanwhile, Professor Anasuya, I have few queries. In fact, I mean, it's a very interesting work which you are doing. Particularly, the interest came in when the interest came in when you talk about both the cosmetics and the drugs or the pharmaceuticals, particularly the cosmetics section.
Yes.
And if you see, in case of cosmetics, there is one label. Mainly the companies want to put it on their cartons that it is cruelty free.
Yes,
and
yes.
you are actually providing an alternative to test their materials or their cosmetic products without going into the animal testing, which is not at all recommended when we go for the cosmetic applications. That's really interesting. Just I want to just know more on what you are envisioning when you are going for the testing sites. Say you are developing a scaffold with different cells embedded into it and looking for a cosmetic application. If you have some product that you have envisioned or the type of a test you have envisioned.
So I guess what you want to know is like in what form I will be giving the product to the company for testing. So there are 2 ways for the skin. One is we can make spheroids or organoids, like 3D small, spheres made with scaffold and cells for testing. And we normally grow them in 96 well plates, like small, tiny Petri plates kind of thing. So that we can give in multiple numbers. So it can be tested several times. and it will be consistent. So that is also very important. So there will not be batch to batch variation. So it will be like identical things to test on. So one is that we can give you spheroids or organoids of skin for testing. And the other is we can give skin patches, like 2D culture kind of thing, like a sheet. So small pieces of sheet on which you can test. But all will be in Petri plate or 9/6 well plate kind of vessels. So in that you can directly add your, just like we do cytotoxicity testing, we can directly add the cosmetics and test it. And
Yes.
like for, yes,
That's really interesting to know if we can have a patch of a
Yes.
skin and where certain
Yeah.
tests could be performed.
yeah,
Particularly the safety assessment is the first one that we always look for any product
Yes.
when we are bringing into the market, be it a cosmetic or be it a pharmaceutical product. So that's really interesting.
thank you so much, Professor Chitkar.
Ma'am, we have a question in the chat box.
Yeah.
So this is Dr. Charan Singh wanting to know how the cells incorporated matches with the actual skin fabric. Secondly, it's mono or combination of cells and which layer of the skin.
Yes. So that's a very good question. So we are currently working on the top layer. So it's the epidermis. And also it is the easiest to work with. So we can have one type of cell. We are working on co-culture, like using two different type of cells and building the full thickness of the skin, the dermis, everything. That's a bit challenging, but we are working on it. The problem is when you add two types of cells outside the body, one tries to outgrow the other, meaning both do not match the same pace of growth. So after a while, you end up with one type of cell, So we are working on it. There are mechanisms in which we can separate them, like put a membrane in the scaffold so that two different type of cells can grow on two sides, something like that. So yes, that is also in the research stage, but that's a very good question. And we use different kind of cells. We use normal keratinocytes also, and we use different kind of skin cancer cell lines that are little easy to work with. So for the testing phase, but we also work with normal skin epidermal cells, keratinocytes, fibroblasts, etc. So it can be customized also. We can grow almost any type of cell on these scaffolds. Thank you.
Okay. Hello, ma'am. This is Charan Singh.
Yes,
As
hello,
you
sir.
explain the question. One more I wanted to ask is, how about the regulatory aspect of the same if you are using the one type of cell in the scaffold?
So firstly, the scaffold is not a problem. So getting regulatory, certification won't be a problem because these are tested. Many of them are pharmaceutical polymers. Then some are natural materials. So I think it'll be not that difficult to get. But in terms of cells, yes. So the thing is, if we are applying it on someone, so then it has to be compatible with that person.
Yes.
Nowadays, what people normally do is they take a biopsy or a small skin, piece from the patient or from the person whom we are going to use it on. And then we can grow cells from that particular biopsy material. And that we can put them on the scaffold so that there is no chance of rejection. But that is for a patient. Now, for pharmaceutical testing and cosmetic testing, we don't need that. We can use any type of cells, and depending on the what kind of tissue you are testing on, so we can customize it with a certain type of cells, and there's no rejection issue in that. So it's just a test. So that is, I think, regulatory certification will be easier for the testing kits.
Mm-hmm. Okay, thank you so much, ma'am.
Yeah, thank you, sir.
Professor
Yes,
Anupama, you have your hands on. Do you have any
yes, Professor.
comments?
Uh, I just wanted to ask the question to Professor Ganguly. Professor
Yes,
Ganguly, I
Professor.
was just wondering, you talked about corn husk-based scaffold. So that would
Yes,
provide a two-dimensional environment for the cells to grow.
yeah,
How does it overcome the requirement of a 3D
yeah,
topography of a scaffold which is required for the cells
yes,
to grow? That's my
yes.
first question. And the subsequent question would be, did you get a chance to test that the cells which are growing on 2D and the ones which grow in 3D, are they exactly similar to each other, not just phenotypically in terms of the fibroblastic shape that they attain, but also in terms of their genetic makeup? Do they show same type of functionality? And then how this 2D scaffold differs from the conventional cell culture plates that we are using, which again offer a 2D environment? That was just
Yes.
out of curiosity, I
Yeah.
wanted to know.
Um, yeah. So the first question was about that scaffold being 2D, right? Yes, the corn husk does look like a sheet, but when you see it under the microscope, the SEM, it is also 3D. It is the cells don't see it as a very flat um, scaffold. So it has different layers to it in the microscope. In terms of the cell is very small compared to the skin, the corn husk scaffold. So one layer might hold several layers of cells under SEM. So that is one. And plus, we can make the corn husk into small pieces. We can mix it with other viscous materials and make it into more 3D, like a bigger 3D structure too. So that's a good idea. So if you want it bigger, then we can put several layers of it or small pieces of it embedded in a second scaffold material or a gel-like material. And we can use it for 3D printing, or we can use it, even manually for wells. So just to get a larger 3D structure for the construct. So that is your question number one. And for question number two, yes, the cells will behave slightly differently because the environment is different, especially in terms of oxygen, availability of oxygen and materials. Because when you are growing it in a 2D, in a flat dimension, all cells are exposed to exactly the same media, the same nutrients, the same oxygen tension, everything. So they all behave very similarly. Now, the minute you put them in a sphere, and depending on the size of the sphere, the cells that are on the surface and the cells that are in the center are not being exposed to the same amount of nutrients, the same amount of media, the same amount of oxygen tension. So what happens is they might try to differentiate. And we also provide different growth medium, different differentiation medium. So it might try to, like as we see in embryology. So how from a few very common cells, like all cells are same, and from that cells they kind of differentiate and develop into very specific organs. So that is a hope also that we try to check. And also at the same time, we always characterize everything. So we will check the genetic makeup and the protein profile of the cells in 2D and 3D cultures. So that is also there for for anybody to see. So did I answer your question, or if you have some more question, maybe
Yeah,
you can
thank
ask.
you. Thank you.
Yeah,
Thank you.
thank you so much.
Anasuya ma'am, you seem to have found collaborators already.
Okay, that's wonderful.
So thank you for your presentation, ma'am.
Thank you so much.
Yes.
Yeah, I'll try to let.
So meanwhile, let me introduce our next speaker, Professor Swati Biswas, featured among the world's top 2% scientists in the year 2019, 21, and 22 in pharmacology and pharmacy, nanoscience and nanotechnology, and clinical medicine. She is a renowned faculty member at BITS Pilani, Hyderabad campus. Her research interests lie in the advanced drug delivery systems and cancer therapy. The lab has developed peptide-anchored dendrimers, tocopherol conjugated micelles, tumor-targeted nanomedicines for enhanced chemotherapy. Additionally, they also worked on sustained-release formulations, epigenome-targeted liposomes, and nanotools for antimicrobial peptide delivery in Pseudomonas keratitis. From having 12 Indian and 2 international patents, let's hear a little more about one of them. Over to you, Professor Biswas. Ma'am, you might be on mute. Can you check this?
Yes. Can you hear me now?
Yes, ma'am.
Okay, thank you. Thank you, Dr. Chitkara, and also the organizing committee for giving me the opportunity to present our research. And of course, thank you, the team. So yeah, so actually we have a couple of patents which are approved. So I will talk about 2 patents which are a little bit different, and that the idea of including another patent come after hearing from Professor Anasuya. So basically, the patent that I'll discuss about is the hydrophilic redox responsive polymeric micelles comprising C6 for photodynamic therapy. So basically, our lab works on various polymeric systems or nanoparticles. The current approach is to develop the nanomedicines and for various disease applications. So we started with cancer in the beginning, and we work on various approaches like chemotherapy combined with photodynamic therapy, or combining with photothermal therapy, or combining 2 different treatments. So all that we start looking into all the efficacies of all that different formulations in cancer. And also another area that we work in the bacterial keratitis, where we develop the formulations which are advanced formulations, and which will take the the beneficial for compared to the conventional eye drops. So we work on various particles and polymeric systems, and also proteins. So for nanocarriers for cancer, we work on human serum albumin, we work on dendrimers or carbon-based materials, which is graphene oxide. For polymers, we work on PLA-based polymer, polylactide or polycaprolactone-based or polylactide co-glycolic acid-based polymers. So all the different systems that we work. So mainly for bacterial keratitis, we actually invented some technology using the 3D printing approach as well. But I would mention that it is not just only 3D printing. We can also use it using the conventional mold method. And so basically the ink, which is important here. And I would like to mention here that there is only one drug-eluting contact lens, which is recently approved in 2022, and that is by Johnson & Johnson, and which actually has an anti-allergic drug in it. And that this is basically done because these contact lens wearers develop this itching, and over the time, like like out of 8 out of 10 contact lens wearers report about this the constant itching. So this basically this Johnson & Johnson developed this AccuView TheraVision, and which has the ketotifen. This is basically that antihistamine drug, and it is for the this contact lens is for the vision correction as well. So for our work that we want to make not exactly like contact lens, but we want to make like a contact lens like patches, and which will be positioned in the cornea, and which will slowly release the drug, and also will be dissolvable in nature. So it will dissolve over the time, and it will give the therapeutic benefits. So the reason that we will have the benefit is the patient will be very much compliant, because it's not easy to administer the drops every half an hour or 1 hour or 2 hours interval, and majority of that goes somewhere and it's not so effective, whereas we can have a local delivery if we make that kind of patch, and it is easy to make 3D printing. And I think it is much better if you use the 3D printing technology, because we can have the right amount of the dose delivered to the patient. So right amount to the patient. Or if it is not there, then it will be difficult to control the dose because it is already prepared. So we have few of the antibacterial study that we have done, and we show that so this, when we use a special type of lens forming material, and then we see that adhering very well and degrading very well, and also it has its inherent antibacterial activity. So we actually proven it with several studies, and it shows that it has the good ability to destroy the live cells in the biofilm, because the cornea actually gets severely infected in bacterial keratitis, and a strong thick biofilm forms over the cornea, and that actually causes the damage to the to the inside tissues. So we have also done some of the studies with the rabbit eyes, and we could see that our treatment could actually help the rabbit to regain this transparency in the cornea, and also bacterial load was minimal compared to the control treatment. So this is already patented technology, but however, as the lab is mainly working on cancer, we have several patents on cancer treatment. So here basically we want to promote an alternate approach than chemotherapy, which is a photodynamic therapy. So photodynamic therapy basically needs the light and also this photosensitizer. So we will make the nano formulation for the photosensitizers, which will actually be delivered via the systemic circulation, and will target the tumor. So that is our main important. And also when I look into the current clinical scenario, then we see that there are only one formulation, which is photofrin, which is in the clinic and which is given intravenously, and which is a conventional formulation where the drug is dissolved in saline and is administered, which has no specificity. And then as there is no specificity, the patient will not be able to go in the light environment, because it will show the toxicity to the whole organ. So because the photodynamic therapy itself can be targeted by using the targeted laser light, so it is basically important to to deliver the drug also in a targeted manner, so that we will have much less toxicity, systemic toxicity. So basically it is a laser dependent process. So we can actually see that they are typically inactive, but when the light falls, they go to excited state and reacts with the biomolecules or nearby molecular oxygen to develop the singlet oxygen, so which actually kills the cancer cells. They are effective, but not so many are in the market. So as I said, that one is in for the cancer treatment, the other one, like the other two are also there. So one is Visudyne, which is for the treatment of macular degeneration, age-related macular degeneration, and where the patient, even though the patient is anti-VEGF treatment, but this actually helps them to stop the proliferation of the cells, of the vascular endothelium. endothelial cells. And also there is one formulation that is for acne treatment, which is using another photosensitizer, which is aminolevulinic acid. So these are the three formulations which you can see in the market, and we see a lot of hope for this clinical translation of this kind of material. So basically, this is, we are using chlorine E6, and this is a structure of chlorine E6. And you can see that this, it is a porphyrin structure, which is giving the ability for it to absorb the light and liberate it at the certain wavelengths. And the good thing about the has these two conjugatable groups which we can modify and make different kind of structures. So how, as I said, that the importance of delivering the drug to the target is very important. And when we you make the normal nano formulations, it has the ability to target the tissues by enhanced permeability and retention effect, specifically due to its size and shape. But and it is very important to deliver it, as because it will improve the safety, it will improve the efficacy, and the pharmacokinetics will be improved because the drug will be inside the vesicles, and which will target the right tissues or organs. Now, for our particular work, we have actually designed the polymer by certain approach. So where we actually developed this chlorine E6, which was modified by polylactic acid and also polyethylene glycol, because polyethylene glycol is something important for the passive targeting phenomena. So we actually, made it by some chemical synthesis approach where chlorine E6 was conjugated, and then we also incorporated the methoxypeg. The reason behind all these modifications were there, we wanted to make the chlorine E6. So this is the structure of chlorine E6. So we wanted to make the chlorine E6 to have the polyethylene glycol attached to it, so that it can form the micelles or self-assembled amphiphilic structure very well. And also we want to incorporate this glutathione in between, so that it can actually degrade and release the drug in the tumor tissue or tumor microenvironment or in the intracellular compartment of the cancer cells. So this is what was the approach, and we have done some of the studies. So the main objective was that whether the micelles are forming or whether the nanoparticles are being prepared. So this is a self-assembled structure, and the hydrophobicity is coming from chlorine E6 and hydrophilicity is coming from polyethylene glycol. And we see that this is the MPEG PLA, polylactic acid, and we could see that our formulation is better in self-assembling. Now, why it would be better in self-assembly? It is basically MPEG PLA, and then we actually included this C6 as a prodrug. Now, why this is important? And because this is basically a hydrophobic, which is actually helping in the self-assembly process. Also, it's very important to know about the particle size, because if it is more than 200, we cannot use it for the tumor, for the anti-tumor therapy. So we have found out that the nanoparticles were in the nanometer size range of less than 200. We also have to find out that whether they are actually releasing helping to release the reactive oxygen species or not. So this chlorine E6 basically will react with the molecular oxygen nearby and will develop the oxidative stress by generating the reactive oxygen species singlet oxygen. So we have several biochemical analysis which was done. The one of them is dimethyl-anthracene, which reacts with the singlet oxygen, and we will see the reduction of their fluorescence because of their inactivity. So we can see that the more C6 is getting released, and we could see, and that too also in presence of GSH, because glutathione is something which is highly overexpressed in cancer. So we have taken that as a targeting approach and to deliver the molecular C6. So we have done even another study with the singlet oxygen sensor green, so which actually determine the level of singlet oxygen. So we see that the very huge surge of singlet oxygen when we are treating them with the our MPEG PLA SSC6 formulation and in presence of 10 millimolar of GSH. So which is basically the concentration of GSH in the tumor cells. So we also perform the cumulative release, and we see that this release is not very abrupt, and it is basically sustaining the release, and which is so important for tumor cell killing because it actually needs the sustained delivery of the drug in the tumor environment. So we actually did some of the other studies also, like in the cancer cells, where we have taken basically the lung cancer cells, and one murine model was taken. So we see that the upregulations are happening. So we could see that these nanoparticles are, getting incorporated. We also checked the viability of the lung cancer cells in after 24 hours of treatment, and we could see it is drastically reducing the cell viability, and this enhanced toxicity was observed in both the cell lines, murine and the human cell lines. So it's not only related to lung cancer, because we have tested it to lung cancer, but it can actually be applicable to all the cancers where the light can go, because it needs the activation by light as well. So the deep tumors may not be a right candidate, but the tumors which are superficial can very well be treated with these photosensitizers. And we also did some of the organoid structures. So we prepared the organoid structures using the A549 lung cancer cells, and to introduce the heterogeneity that the natural tumors are having. So we could see that our formulation was able to penetrate deeper in the tissues. So the reason that it could penetrate more even compared to free C6 and the other nanoformulation is because the nanoformulations have the better penetration. The nanoformulations are basically the stimuli sensitive, so it can basically release the C6 much better way, and also it has a better more hydrophobicity, so that actually helps them to penetrate through the spheroids or go to the intercellular compartments. So this is how important, and we could analyze it with the flow cytometry models as well. So here we did a quick study to find out that if these nanoparticles are reducing the spheroid volume, and we see that compared to control, which is highly proliferative in nature and reaching to 900 micron diameters, but we could see that this one basically suppressing the tumor growth. And now how it is suppressing the tumor growth, we actually started evaluating it, and we found out that there is very less amount of these live cells which are present, but compared to that, we see a lot of dead cells which are present by the specific propydium iodide staining, and so this is how we got the picture. So here basically no glutathione was used, but this is the normal environment of the tumor spheroids, which allows us to assess it properly, and the level of glutathione is already present there compared to the monolayer cells, and which was the limitations for the monolayer cells. So we also checked that apoptosis, how much basically the cell death signal is there in the spheroid. So we see that huge cell death signal in our final formulations compared to the free C6 treatment. So with this, I'd like to conclude. So I think that if you have any questions, please feel free to ask me. So these are the people who worked in the lab, and we have a couple of fundings from different projects, ISC STAR projects, ICMR, Sun Pharma, Dr. Reddy's, DST, ACRB. So all these funding agencies have generously funded our projects, and we also have the collaborators who work actively with us in our projects. So thank you.
Thank you for the wonderful presentation, Professor Biswas. Seems like a very interesting work.
Thank you.
The floor is open for questions if anybody wants to say something.
Hello.
Hello.
Yes,
Yes.
Professor Jagpreet, yes.
Thank you, Professor Swati, for a wonderful talk.
Thank you.
Yeah, I have one or two questions. First one is that you are talking about the photodynamic therapy. So in this, what were the excitation wavelength for this C6 toy?
Yeah. So for these, we use the 633 nanometer wavelength, which
630.
is, which is in the, you can say it's a near infrared. Yeah.
So can you comment over its penetration power?
Yes, so that's why I said that it will be very good for the superficial tumors like head and neck tumors or skin cancer and breast cancer, where the light can penetrate and it doesn't have to penetrate a lot, like not like pancreatic cancer or liver cancer in the other parts. So those may not be, but which can be treated. And also the chlorine 6 is one of them, which has 633, and this is a higher generation compared to the one which is photofreen, which is already there in the market. So this is the higher generation. So the penetration is better than what is available in the clinic.
Yeah. So one more thing. So is it only for the PDT or for the imaging also?
Say it again. I did not understand.
I'm asking, is it only for the PDT or for imaging also?
Okay. Yeah. So that's a good question. So basically, yeah, it is. You can use it for PDT, and as it has its inherent fluorescence, so you can also track it in the imaging.
So it is not showing a switching on or switching off phenomena. It remains in fluorescent. Am I right?
Yeah, it will be fluorescent. Yeah, it is fluorescent. Yeah.
Okay. Thank you so much, sir.
Thank you, Dr. Jagpreet.
So I have a quick question, Professor Swati, for you. It's a really nice piece of work which you have done and patented as well. My question is slightly different. On let's say this therapy has to come out. I mean, your work, we are having a nano system, the E6 conjugated to a polymeric backbone. So how do you envision its future path? Say if it has to be come into the clinic.
Right. Yeah. So as you can see that the clinical status is very poor. So it has only the drug dissolved in saline. So that's what it is at this point. Now, what we are as academicians, what we are thinking it way ahead than you can even think what is in the clinic. So if you have to think about a translation, we can start with normal micellar systems, and which will definitely improve the solubility and targetability. Then we can go for something which can be actively target and which can, but prodrug approach will definitely be beneficial. So we can and it is available also. So I think that in the if you think about the industrial advancement, so that is way lagging, or I will say that it is very slow compared to what we are thinking and going ahead in our research.
Okay. So thank you, Professor Biswas.
We can take one more question if anybody has.
Yeah, we can.
Okay, Professor Swati, I don't think anybody has a question at the moment. If they do in the future, we will let you know. Acha, there is somebody. There is Dr. Charan Singh wanting to know how about the aqueous humor washing of hydrophilic drug in the eye?
Yeah, so that's what. Always the eye drops will carry the hydrophilic drugs only, so it will be washed off. But when we are making these patches, which will stick there, it will slowly release the drug, and the drug is immediately available in the cornea. So it will be the more chance of it going to cornea than hydrophilic drugs washing off using the eye drops.
Yes, Dr. Charan.
Hello, madam. What about I wanted to ask about the efficacy of the same formulation when we are taking into consideration a hydrophilic drug in eye drop or the formulations vis-a-vis antibacterial activity.
Yeah, actually, we have seen that. We have the animal model also that confirmed that basically we used a peptide for the first study, but then the peptide was water soluble. So when we gave the peptide, it was not effective. I mean, it was effective, but not significantly different than the control. But when we had our formulation, like when we put the patch on, so that drastically actually increased the therapeutic efficacy.
Okay. Good. Thank you, ma'am.
Thank you for asking your question, Dr. Charan. Uh, Professor Swati, we'll move on to our next speaker. Thank you for your time. It was a wonderful presentation.
Thank you so much for the invitation. Thank you, Dr. Chitkara. Thank you, Anjali. Thank you.
Thank you. Finally, introducing our last speaker of the day, Professor Anupama Mittal. Being the convener of Institution's Innovation Council, Professor Mittal is a faculty at BITS Pilani campus. As full of energy she is, so are her research interests. She is keen to explore multiple fields, including but not limited to nanoparticles and polymeric micelles for site-specific drug delivery, polymer or fatty acid prodrugs for effective treatment of cancer and diabetes, stem cells and exosomes as biogenic carriers of miRNA and protein in cancer, and growth factor and peptide-based therapeutics for wound healing. To having 4 patents granted and 7 more filed, let's hear a little more about her innovations. Over to you, Professor Mittal.
Thank you, Achal. I hope I'm audible to you
Yes, ma'am, we can hear you.
and you can see the screen as well.
Yes, ma'am, clearly.
Thank you, BITS Tech, for providing this opportunity. And thank you, my earlier speakers, Professor Ganguly, Professor Biswas, for the wonderful talks. So now I'm the last person, and I have this responsibility of keeping the interests of the audience intact. So today, what I'm going to talk about is a very specific anti-diabetic formulation. This project was sponsored by DST Serve for the initial part, and 2 patents have already been granted on this technology. So one of the major segments of our research group, of our research interest, is working on diabetes and diabetic complications, wherein we are delivering small molecule drugs like lisophilin, about which I'm going to talk today. And we are working very actively in peptide delivery. Some 3 patents have already been filed on these peptides. And also we are working on regenerative medicines for diabetic wound healing. Today, I'm going to focus on one of these technologies, that is how we made a drug which was very challenging to deliverable in the form of tablet. And this screen shows my entire journey of this particular project work. So we started here with this molecule lisophilin. And the problem with this molecule is it has a very short half-life of 8 minutes in the body. And that is why it has to be given by infusion, by injection for 8 to 10 hours clinically. And the problem is so severe that there is hardly any research effort, any literature reported on this drug towards its clinical translation. So when we started and we were funded by DST, my only objective was somehow to make this drug have a greater residence time in the body. And the first step towards it was making a prodrug and using a fatty acid. But then, because we wanted it to become clinically translatable, we wanted to have oral delivery. We encapsulated in some specific polymer which could be available orally. And further next, based on the success of what we observed with this polymeric system of this prodrug, we further went ahead to convert it in the form of tablet, keeping in mind the industrial viability and the patient compliance. So this work has already been granted 2 patents and 7 publications have been there on this single work. And the work was also cited by Nature India. It has also been recognized
Hello.
by the Sun Pharma Science Foundation. So now diving into the research aspect of what was done with this molecule, this is the molecule, and it is the presence of this OH group in this molecule, which results in a very quick metabolism. And this snapshot shows how it metabolizes into its parent drug pentoxifylline. And because of this quick metabolism, it requires an infusion of 8 to 10 hours. This drug is primarily immunomodulatory and anti-inflammatory in nature, reported for several disorders, primarily diabetes. So for a chronic disorder like diabetes, wherein the patients require at least 2 times the drug or the tablet has to be taken or insulin injection has to be taken, we cannot even think of a drug which has to be given by infusion for 8 to 10 hours. So that is what generated our interest in the molecule. And the way we started was we first wanted to shield this OH group by making a prodrug. And then we thought of delivering it orally. So I'll take you further with this stepwise. So the first step was we selected a fatty acid, linoleic acid, which was selected based on being an essential fatty acid and that it would be beneficial to diabetic patients. And then the molecular weight was balanced in a way that the prodrug that we got, interestingly, it was able to self-assemble without using any surfactant into micelles. And the critical micellar concentration that we obtained was much lesser than what is reported for many of the commercially available surfactants. So the first patent that has been granted on this project is mainly on this innovativeness of getting it self-assembled without using any surfactant. And thereon, we performed pharmacokinetic studies wherein we saw that there was a 5-fold increase in its residence time in the body. Even when it was given in rats, in diabetic rats, what we observed was clinically, the dose that was given for free drug was 25 mg per kg twice. And when we started with that dose, we found that the rats became hypoglycemic. So the effect was that much. And that that compelled us to reduce the dose from 25 mg per kg twice daily to 15 mg per kg once daily. So there was a reduction in dose as well as dosing frequency. And that is what you see here with the red plot, that from the 5th dose, we got a stabilization of glucose levels, which was much better than free drug or the drug being given at a higher dose with a BID frequency, that is twice a day. Therein, of course, we measured the insulin levels and insulin cells were also stained. So we got a better response as compared to the diabetic control free drug and the higher doses one. Then we went ahead to see now this LSF-LA self-assembled micelles that we have prepared. Are they available orally? But that was one of the major failures that we encountered. When we tried giving it orally, we did not get much bioavailability. It was just 24%, which was almost similar to free drug. As you can see here, the first 2 graphs are for the free drug as well as for LSF-LASM, and both showed just a bioavailability of 24%, which became very disappointing for us. And we were fraught with the question that what to do next. So next, what we did was we synthesized a polymer in the lab, and the polymer was able to entrap this LSF-LA polymeric micelles. This is nothing which is very innovative. This is reported in literature. These type of polymers are being prepared and such type of drugs are being encapsulated. But yeah, we encapsulated prodrug in our case. But interestingly, the interesting part was, I just told you that orally, when we tried to test the LSF-LA self-assembled micelles, we just got a bioavailability of 24%. But same thing, when we tested orally with this lab synthesized polymer, the bioavailability increased to 74%, which was 3 folds of what we observed with naked micelles. And herein, our objective was not to increase the potency or efficacy of LSF. That we already attained with LSF-LA self-assembled micelles. Objective here was to make it orally bioavailable, wherein we did observe a threefold increase in oral bioavailability. And just to confirm that it retains its potency, we checked it in a diabetic model, wherein we found that LSF-LA polymeric micelles, whether we administer it intraperitoneally or orally, or just LSF-LA self-assembled micelles intraperitoneally, all of them brought the glucose level to the same level, which shows that the potency of the conjugate of the prodrug was intact after encapsulating it in the polymeric micelles. And further next, if you see this graph, even the insulin levels were significantly increased and were similar to that of LSF-LA self-assembled micelles given by intraperitoneal route. So herein, the major outcome was we were able to get the oral delivery of this conjugate. And further next, we wanted to deliver it something in a way which is more patient compliant. And we know that one of the best patient compliant and industrially viable dosage form is tablets. We selected a particular array of excipients, and with those excipients, we prepared lyophilized powders and which were compressed into these tablets. So this is how we were able to get the tablets. But now the obvious question was, because we have compressed it into tablet, do we still have those nanoparticles with which we can harness the oral bioavailability? And therefore, what we did was these tablets were made to undergo the disintegration tests, which are recommended by any official Compedia for tablets. And after disintegration, we collected the media that we got. And after a few centrifugations, the pellet that we got, we measured its size, and the size was same as that of the initial nanoparticles, which were compressed or which were compacted to get these tablets. And thereafter, only in that pellet, we tried to check the amount of the free drug that was present, which was encapsulated in it. And indeed, we got similar amount of drug which was added initially into the tablets, which confirmed that even after compaction into tablets, the LSF-LA polymeric micelles were intact. Just to check whether they are orally viable or not, we perform single-pass intestinal perfusion studies, wherein we measure permeability coefficient. And therein, if you see these PLM in the form of tablet, they had almost twofold higher permeability coefficient as compared to LSF-LA self-assembled micelles. And we also did the pharmacokinetic evaluation of these tablets in rats. So in rats, we cannot give tablets. So what we did was, that was an experimental limitation. We had to crush the tablet and then it was administered into the rat. But the interesting part was we were able to still observe this curve, which you see this as orange plot, wherein the drug did show the plasma concentration in rats. And this is how we were able to convert this very hydrophilic molecule. It is so hydrophilic that it has a solubility of 60 mg per ml in water, and it has to be given by 8 to 10 hour infusion, and how we were able to convert it into tablets, which are very easy to be administered to patients, which is an industrially viable option, and it did show the effect in vivo as well. So talking about the translational potential of this technology, its value proposition would be mainly what I have been saying, that it involved molecular optimization of this drug into an orally active prodrug, which was assembled into polymeric micelles and then compressed into tablets. Our target customers would be primarily diabetic patients who are suffering from type 1 as well as type 2 diabetes. Not only that, diabetes does not stop at that stage and it proceeds ahead to diabetic complications. So it could even work good for diabetic complications. And it has also been evaluated in our lab for diabetic nephropathy, wherein we have been finding very interesting and promising results with the same formulation in diabetic nephropathy. Currently, our technology is at TRL4. And next, what we plan to do is toxicity studies of this formulation in a GLP certified laboratory. Innovativeness lies in our multi-step approach. of making this drug orally bioavailable, and that too in the form of tablets. Yes, there would be potential threats to the market, primary one being that for type 1 diabetes, the mainstay is insulin-based therapies. Although there has been a lot of hue and cry about non-insulin therapies because daily insulin injections are very inconvenient and they lead to injection site reactions. But over a period of time, it is also possible, because diabetes keeps on proceeding ahead and progressing, that this technology may just serve as a adjunct therapy to insulin in later stages of diabetes. But competitive advantage is what we feel is it being orally deliverable, and we hardly have any therapies for type 1 diabetes other than insulin-based therapies. So 2 patents have already been granted on this technology. And the addressable market size, if I talk about, it's huge, as all of us know. In fact, just talking about type 1 diabetes, globally, it has been set that the market size for type 1 diabetes is 7.59 billion, which is envisioned to increase 13.64 billion by 2030. And the best part about this technology is, although we have just evaluated it in diabetes and diabetic nephropathy, but because it is an immunomodulatory drug, it is very well suited for other autoimmune disorders, particularly organ transplantation, including islet transplantation, again for diabetes, and maybe for other autoimmune disorders as well, like rheumatoid arthritis. So with this, I would like to end my presentation. Thank you all, and I would like to acknowledge my PhD students who have been working with me on this or other projects. Thank you all.
Thank you for your wonderful presentation, Professor Mittal. It seems like a very innovative solution to a very common problem that a lot of people face. Like it's a very basic, keeping a track of how much tablets are you having at a particular time, that if somebody is having a lot of medication prescribed to them, that can become a task. Okay. The floor is open for questions if anybody wants to ask or say anything. Yes, Dr. Saran, please, you can unmute yourself.
Hello, ma'am.
Hi, Saran.
Ma'am, it's very nice to see the current research work going on in your lab at Pilani. And if you allow me, I can speak in Hindi then. क्योंकि इतने टाइम बाद आपसे इंटरेक्शन हो रही है, and I can recall the time when we were in the same lab during your PhD and my masters at Naipur Mohali. So,
yes, I do remember, Saran.
ma'am, this is the quantum of work you have presented here through this BIS Tech, and I'm very happy to see the outcomes of the work in terms of IPR also. And at the same time, you are touching the state, that particular state of the society, and our dear India is particularly known as diabetic capital of the world as well. So far, your current research work is concerned. So you are touching that particular segment, or the population of our dear India in terms of this particular novel formulation. And my only question is regarding the use of lysophilin in psoriasis. Like you have explained about the use of the same drug for autoimmune disorders. So can we explore this particular molecule in psoriasis also?
I believe that it should work in psoriasis, although I have not done it in my lab, but because it is a very well-known immunomodulatory drug, I believe it should work very well in psoriasis, and psoriasis just involves a topical application. So maybe just the direct prodrug in the form of self-assembled micelles should give a very good efficacy in psoriasis. We may not even need to go into the polymeric formulation. Just the first step, what's written here over here as A, should work very well in psoriasis, Charan. That would be a good idea to explore it in psoriasis.
Okay. Thank you so much, ma'am, for your wonderful presentation and making the doubts clear. Thank you so much, ma'am.
Thank you, Charan.
Professor Ganguly, you have raised your hand. Probably you have something.
Yeah, it was a wonderful presentation, Professor Mittal. Uh, yeah, I'm not much aware of the chemistry of these molecules. So I was just wondering how one drug is helping in diabetes plus nephropathy and autoimmune disorder and psoriasis, or they're all different molecules, different
No, it's just a
compounds.
single molecule.
Okay.
So first part that you asked was diabetes and diabetic nephropathy. So diabetic nephropathy is a complication that develops subsequent to diabetes. And the major reasons for development of diabetic nephropathy is persistently high glucose levels. Although diabetic patients are on anti-diabetic medications, but as we all know that there are spikes of glucose level in the blood, and when it happens over a prolonged period of time, slowly and gradually, the person also approaches towards nephropathy. So, lisofilin in this case, it helps in keeping the glucose level under control, stabilized, and that is how it delays the progression towards diabetic nephropathy. So that's about diabetes and diabetic nephropathy. Coming to the second part about the autoimmune disorders. So lisofilin is not just an anti-diabetic drug. It's that we are using it for diabetes. Actually, it is classified as an immunomodulatory molecule. It modulates the immune system. It suppresses the immune system, and that is how it prevents certain autoimmune diseases. So that's the reason we say that it has a broad spectrum of activity, which may include diseases or disorders like psoriasis as well. So I hope I've answered that question.
Yes, thank you, Professor Mittal.
Thank you.
Thank you so much.
So, thank you, ma'am, for your query. And again, we see more collaborations within the meeting itself. Right. Any more questions? If anybody has, kindly let us know. So we haven't heard from anyone as of yet, Professor Mittal. If and whenever we do, we will reach out to you. So
Sure.
thank you for your presentation. It was very informative. So with this, we are done with all the presentations of our faculty innovators. But before we conclude our today's session, we would like to give our audience.
Actually, sorry to interrupt you here.
Yes.
May I stop sharing?
Yes, ma'am.
Okay, thank you.
Okay. Okay. So before we conclude our today's session, we would like to give our audience a very small glimpse of the TEC's resources and services that we think will be helpful in assisting everybody in their entrepreneurial journey. So with this, I invite Mr. Sahil Sheikh, our IP and Technology Officer, to elaborate a little.
Hello everyone, I hope you enjoyed the overall session. So before I show you what are the resources available for you guys, I would just like to elaborate why we have done this, because we are from DST, and one of the mandate of DST Tech is to create one platform where all the stakeholders can come together and explore different resources like IPs, what are the research opportunities are going on in the state. So because of that, we have created a kind of platform where we are trying to make a landscape of the state of IPs and researchers. So I'd like to show you this Rajasthan IP dashboard. As you can see in my screen, this is kind of a data IP landscape of the state where you can do search. We have mined different IPs from the different assignees. So you can search by the assignees, like different assignees are here. You can search by technology domains also, we have categorized. You can search by year-wise as well as IPC, and you can put the inventor name also. And if you are particularly looking for any particular domain, then you can just type that something keyword related to that. So if you do that, all the IPs relevant to that keyword will come up. Just you can see here. So all the solar technologies will be available, and you can read, and you can, if you are interested, you can foster collaboration, and you can go ahead with that. So similarly with that, here you can see the IP landscape of the state. So how many technologies right now available? What are the percentage of share of different technology domains? Then you can go ahead and you can explore further trends in the state, like how many patents are getting published in a year, what are the legal distribution, who are the top innovators in the state. So all of the things you can discover here. And if you want to do further exploration, if you want further collaboration, you can connect with us. You can write to us, then we can initiate further collaboration. As well as, similarly, we have created another database, that is Rajasthan faculty database. Basically, if you just put any inventor's name here, like if I'm putting Deepak sir's name as, so you can see, so number of the professor would come with that particular name, and then you can like explore more about them, about the researches, about their particular specialization, and you can foster collaboration. So these two databases, I think, would be helpful for everybody. Like you can be a student, you can be a startup enthusiast, researcher. So these two databases can really help you. This is available right now at our BITS Tech website. You can go here and check the databases, and you can like explore these databases.
Thank you, Sahil, for highlighting our efforts in hopes that it will be helpful to anybody and everybody out there. So with this, we conclude our first episode of TechMate.
Yeah, just
Last words.
Okay, so it was, I mean, really nice to have all the innovators. And one thing you might have noticed that we have all the women innovators today. They are really doing great. And, you know, I look forward to foster further collaborations among the community. And as I said initially, that one of the mandate of TEC is to create that an ecosystem where the technology translation can happen. And translation, for a technology translation, we need a technology. And then various stakeholders come into the picture. So BITS Tech is very open to these collaborations, and we love to hear from you all. You can anytime reach us. If you need any support, we have a team, dedicated team, who can fill in different, you know, components. So looking forward to have much more collaborations in the near future. With this, I thank everyone, the speakers of the day, and all the participants for joining in. So with this, we can now close the session. So thank you everyone.
Yeah. Thank you all. Thank you, Dr. Charan, also for thanking us. It was a very nice, get together of sorts with technology and everybody else. I really enjoyed it. Thank you so much.
Thank you, ma'am.
Thank you, BITS Tech team, all the participants, and thank you, Professor Ganguly.